Hello everyone, welcome to our Oceans Day special. Today we have Yasmin Meeda. She's a postdoctoral researcher at the Cranfield University. She works at the intersection of synthetic biology, metabolic engineering, and water systems. And she focuses on developing genetic tools for heavy metal recovery and engineering biofilms to enhance biodegradation drinking and wastewater systems. She did a PhD at University of Exeter, where she used CRISPR-Cas9 gene editing to investigate phosphorus calcium signaling pathways in marine diatoms, which was vital to understand how marine life responds to environmental change. She also holds MRes in marine biology from the University of Plymouth and a bachelor's from the University of Brighton. And she's also very famous on Instagram. Yeah, there's the link on our Luma page if you're interested. Yes, go ahead.
Perfect. Thank you so much for attending and happy World Oceans Day. So today's talk will be less about the specifics of my research, but more about what engineering biology means for the environment, how we're working towards achieving that, and a little bit about the research that I'm doing and touching on a little bit of policy. So that should lead into the panel this afternoon really nicely. So I titled the talk, Can Synthetic Biology Help Clean Our Waters? And we're looking at engineering biology solutions for emerging pollutants. So at the top here, it says EBIC, Environmental Biotechnology Innovation Centre. I'm part of this hub across the UK. We are 10 universities or institutes, and we're all working to try and solve the problem, I say solve, of pollutants in drinking and wastewater. And this is an image of me with a 200-litre bioreactor. And I recently went snorkeling in Japan, and I'll come on to a bit about my Japan trip. But for World Ocean Day, I thought I'd make it a little bit relevant. So let me go into this. From a really nice introduction, I'll just kind of recap that. I did my work on diatoms, looking at phosphorus sensing and calcium signaling. And this was really fundamental research. And while I loved that aspect of the research, I wanted to get into more of the applied route. And that's what led me into my role now, where I'm working directly with drinking and wastewater companies to try and implement synthetic biology. I also use social media as a way to communicate science. And lately I've been getting into policy a little bit more because science isn't standalone. We really need to talk to the right people and get our science out there. So Cranfield University, it is a UK postgrad only university. So not many people have heard of it. We are based about an hour north of London. If you can see this cursor here. So we're based here with the pin drop. And we're quite a unique university. We have our own airfield because we're an ex-military base and people do a lot of research on aerospace. But one of the unique features are that we actually have our own drinking and wastewater facility on campus, which makes us really great for studying synthetic biology or in general, drinking and wastewater treatment. So you can see here is our wastewater facility, and we've got these trickling filters, and we also have a drinking water pilot scale facility, allowing us to really scale up. And I'll get onto what scale up means a bit later. And so World Ocean Day, reminding everyone to think about the big picture of what we do as researchers. And there's some statistics that state that 80% of marine pollution comes from land-based activities. So everything starts upstream. And I moved away from pure marine biology, looking at diatoms in the ocean and how they sense phosphate, to more upstream. And I'm focused on the discharge from fertilizers or other pollutants that come from land-based activities, and how that gets into drinking and wastewater facilities, and then how that gets discharged. And with wastewater facilities, after we've cleaned up the water, that can get discharged into a local brook, and that's once it's cleaned up. But if there are any contaminants that are hard to remove or aren't removed from conventional processes, this can end up in the ocean. So throughout this talk, just remember that the waterways are connected and how we always relate to the ocean, but I think it's more aquatic biology. And there are many emerging contaminants that have become problems. Some of them people probably don't even think of, We have pharmaceuticals, such as any medications that you're taking, even any medicinal creams. They all come off in the water system. Where do they end up? They will end up in your wastewater system, or they can also end up from runoff going into aquifers for drinking water or the groundwater. So you really need to sort of be conscious of what you're putting down the drains. Another one is personal care products, your shampoos, your skincare. All of these might have chemicals in, and sometimes they're really hard to remove any of the contaminants that are within these products. Another one that's actually emerging is DEET. So when you use insect repellent, that's actually become a bit of a problem within our water systems because nobody really knows how to get rid of it. And again, I've included pesticides, which is kind of the focus of what I'm doing at the moment. In my bio, it says heavy metals. That's part of EBIC and the work we're doing. I'm kind of focused on several different projects all at once. And there are also nutrient pollutions, which I will also come on to. So I've kept my phosphate story from my PhD, and I'm looking at wastewater treatment with that. But you can imagine that different nutrients are applied to crops through fertilizers. This runs off and ends up in our water systems. This will cause eutrophication, which is where there's a buildup of excess nutrients. Algae cause algal blooms, so they can grow exponentially, and they can actually block the sunlight and the oxygen from the water column. This leads to oxygen dead zones in the ocean, and this is really problematic, especially there's a large economic loss because of fish stocks. And if you think about aquaculture or fishing, that can be really detrimental to the economy as well as loss of life and biodiversity. So a quick overview of drinking water treatment, as I'm not sure what everyone's background is on, but it's just to understand that water can come from different places. Sometimes you can have wastewater treated into a river, and then that can be used for water treatment. And then you can also have groundwater, and a lot of the pollutants can enter groundwater through the soil and then into groundwater. But this example is of a slow sand filter, which is a microbial process. So we can apply natural wild type microbes and pass the water through, and the microbes can naturally degrade different pollutants from the system. We also use activated carbon. It's a form of carbon that can absorb different pollutants onto the surface and get rid of them that way. So they're kind of some of the conventional methods that we use. But one of the focuses that I'm using for the synthetic biology aspect is more of a proof of concept. So we're looking at metaldehyde. It is a pesticide, or more specifically, a molluscicide. It's been used to kill slugs and snails. It was actually used up until a ban in the UK, which was implemented in 2020. And this is because of toxicity levels to the environment and knowing that we shouldn't be using it. But obviously, it's been used for many years before that. So it's a legacy pollutant. It's in our soils and it can get into our drinking water. And it's very hard from the conventional water treatments to actually remove it. So activated carbon isn't great at doing this. This map just shows a couple of the catchment areas of where metaldehyde has been found from an analysis done in this paper. So again, this is a safety data sheet that I've taken from Thermo, just showing you some of the hazard warnings of what metaldehyde is and what we're working with. And the main thing that we relate to when we're working with water systems are what are the regulations within different areas. I'm based in the UK, so we're looking at UK regulations. We've observed metaldehyde, this pesticide, at levels 10 times higher than what the regulation limit is. So the regulatory limit is 0.1 micrograms per litre. However, we've observed in the catchments previously shown, they were above 1 micrograms per litre. And we really want to use synthetic biology or engineering biology to help remove metaldehyde from our systems. And I guess nature has a way of doing things naturally, and that is absolutely brilliant. I won't go into the pathway too much, but this is the breakdown of metaldehyde that occurs by 3 essential genes for this pathway that break down into a non-toxic form of acetate. And this gene cluster can be found in some natural occurring microbes. So we have an Acinetobacter species and a Syngobium species that were obtained from contaminated soils that had metaldehyde in. And we know that they can break down metaldehyde through this pathway. So what you can see here from the blue line on both of the graphs are the concentrations of metaldehyde, which you can see on this axis here. And while you get enhanced growth, which is measured as OD, you can see that metaldehyde drastically drops within a day or 2 days for each of the species. So again, this looks great. Nature can solve it. We can use microbes. However, science is sometimes a little bit more complicated than that. And this is an example of the wild type Acinetobacter species. And we have the gene cluster, but it's present on a plasmid rather than integrated into the genome, the chromosome. And so what can happen is when we lose selection pressure, if these microbes are grown on normal drinking water that doesn't have the presence of metaldehyde, they actually can lose this plasmid. It can be expelled from the cell, and therefore the activity of breaking down metaldehyde will stop. So part of EBIC, what we're trying to work on is to engineer this for safety reasons. We need to actually think about the way that we engineer our strains. If we think about horizontal gene transfer, if we add in a plasmid-based overexpression line, this could potentially go into other organisms that naturally occur in our system. And then we might not have a way to control the system. It could be that we've got engineered microbes entering the natural environment. So one way that we are looking to implement the synthetic biology is through chromosome or genome integration. And working with one of the collaborators, Newcastle, part of EBIC, they have used bioinformatics to identify neutral sites, so different flanking regions, either side of a pseudogene, where when we integrate into the genome, there shouldn't be any interruption to normal gene function or to any of their physiology. So that's a computer-based approach, and then we are testing to see if we can insert our overexpression gene cassette to see how that's going to work, and hopefully it does. So we have an approach that we're using as part of EBIC, which is a SAC-B counter selection. I guess I will assume people have a knowledge of genome engineering, as it's a SynBio audience, but if not, it's a Golden Gate strategy where we can introduce an antibiotic cassette into, we integrate into the genome, and then we can counter select on sucrose. So with SAC-B, it will become toxic to the cell and kill the cell. Whereas if the cell has undergone a secondary combination and sort of got rid of the antibiotic resistance and the SAC-B, then the cells will be able to grow on sucrose. So we can then we'll be able to get our markerless mutant. So we don't want any antibiotic resistance genes in there because we're working with environmental strains. We don't want the possibility of antibiotic resistance genes getting into the environment. So that's the approach that we're using. And this is the plasmid-based approach that we are doing. But one of the challenges are that we're working with non-model organisms. We aim that if we're using wastewater treatment, we want microbes that are environmental isolates that can actually survive and cope in these conditions. We don't want to just add in E. coli or a model organism, because sometimes they'll just die and be outcompeted by loads of different organisms. So we're really trying to think of the ecology, and we're trying to understand how we can implement the best strategy for survival, and then obviously ultimately break down a pesticide. So again, we need to think about, we don't have any tools for certain non-model organisms, especially environmental isolates, and the process can be really long-winded. So we need to sequence the genome first, we need to annotate it before we can even get into trial and error on our processes. So we do have an alternative strategy for scarless integration, where we don't want antibiotic resistance and we want the gene cluster to be integrated into the chromosome. And that is this FES-S, and that's based on a paper here. So we're kind of working on two different streams because sometimes SAC-B doesn't work very well for a lot of different bacteria strains. And we're using electroporation as our mode of transformation for most of these, but we can also use conjugation where we can actually get the plasmid into the cell using pili. So there are different approaches that we have to work through, and I guess that's quite a challenge in itself. So I'm currently still working on the two different strategies for genome integration for the metaldehyde structure. But as metaldehyde was banned in 2020, people may be thinking, what's the point? Obviously, we've still got some presence of metaldehyde in our system, but what we're really doing is using this as a proof of concept so that we could then implement this strategy across different pesticides. So it could almost be a plug and play system. Once we've understood what we need from a more holistic approach, what strains we need, what genes are responsible for breakdown, then we can look into doing this on a bit of a more broad scale. And so I've mentioned that Cranfield has a drinking and a wastewater facility. And so drinking water is only part of the problem upstream of the oceans and thinking of how we're cleaning our water systems. And so wastewater can be discharged into the river. And I've put once cleaned because I don't want people to sensationalize what they may have seen in the news where there are clear headlines that have been appearing recently. But there are various different systems that go that are on a wastewater treatment plant, and lots of different cleaning happens on each of these. And here you can see the trickling filters like what we have at Cranfield. But once cleaned, it does get eluted into the river. But sometimes it can be really hard to use conventional methods to get rid of some of these emerging contaminants or some of these nutrients, especially down to the regulations that we need. And obviously, I mentioned that I did phosphate within my PhD. I've clung on to that a little bit. I think it's become a bit of my baby and what I've been working on. So excess phosphate can be present in wastewater, and this can be slightly problematic because it can enter from different sources, whether that's from agricultural runoff or from weathering of phosphorus-rich rocks, and this can enter our water system. And the UK regulations have actually stated that by January 2028, we need a 50% reduction in phosphorus, which is going to be really challenging. So we need to come up with solutions where we can actually get down to the regulatory limit. And just a quick mention again about the oxygen dead zones. So this is when you've got the fertilizers entering the ocean and into coastal areas, and this can cause algal blooms. And that was kind of the focus of my PhD, looking at how these systems respond to phosphorus. And this can be an incredibly big challenge. And so what we're doing at Cranfield, another project that we've been working on is using microalgae. We're actually using a Synococcus, so a cyanobacteria, to try and engineer the strain to be able to remove excess phosphorus from wastewater. This is a setup at lab scale in flasks, and from one of the papers that identified this strain, we're using the PCC 11901 strain. They identified absolutely phenomenal, I think, OD over a couple of days. And so we've identified this really fast growing strain, and we want to remove phosphorus. And one of the challenges are, again, we really need to make this translational to our research. So can this strain that actually is a marine strain originally, how can it grow on wastewater? It's fresh water. It's got loads of other things in there. There are loads of other microbes. You will have competition. You will have different nutrients. You might have things that are limiting. It's not the same as working with a nice lab media where you know everything grows. So we've done a couple of tests to see how we can, if the wild type can actually grow on this. We've got controls on both of these images. We've got PCC standing for just the strain itself. And we wanted to test to see if we didn't autoclave the water and just used the wastewater. And you can see the wastewater is actually quite clear. So this is what we call final effluent wastewater. It's been through pretty much most of the treatment. And we actually saw that after 7 days, there was not much growth at all. Not that you could identify when we didn't autoclave or sterilize the wastewater. However, when we autoclave the wastewater, after 7 days, we saw growth, which is probably because we've killed off any of the competition. And so it's allowed growth. And then we actually just left this as a bit of this probably didn't work. And we left it static, not shaking in our normal conditions. And after 15 days, there was growth eventually. So it was a lot slower growth just in flasks with the non-autoclaved wastewater. So again, when you're doing synthetic biology and you're doing it with real world systems, you need to think of a lot more than just, does it grow in the media and how we can translate this? And what we wanted to do is first we needed the baseline of how does a wild type react? How does a wild type take up phosphate? And we did controls with and without gas. Just we were pumping in 5% CO2 and then compressed air to help them grow because they're photosynthetic. They require CO2 and to produce oxygen. So what we saw is when we had them with the strains with pumped in CO2, they actually did decrease the phosphorus levels down to about 2 milligrams per litre. However, that is not good enough for what the current regulations are. And again, our goal is scale up. EBIC is all about scaling our systems up into real world systems. We can't just keep working at lab bench and thinking, we're going to solve the problems of the world. So this is a 4-litre photobioreactor, and we've been testing these systems in this bioreactor. And then eventually, once we've got our markerless strains through the SynBio, we'll be able to actually scale up with this and use the engineered strains. And I guess what we really mean by scale up is that there are many, many challenges. And I think we'll kind of come onto that in the panel later. But what does scale up mean to everyone, I guess? There's a 7-litre bioreactor here. This one's a 300-litre bioreactor with a max volume of 200 litres working capacity. So this is great if you want to produce high value products, but for wastewater treatment, how can you keep a system that's continuous? Or even for drinking water, how are you going to grow the microbes? Do we need kill switches? So kill switches are fundamental, or we think it's fundamental in microbiology to be able to induce an off button to actually kill our strain. But when we think about kill switches, we actually get into this controversial area where we need to build the public trust. If we want to implement these systems for people, not just in the UK where I'm working, but if it's a global scale, and you tell local people who aren't biologists, we're using GMOs, genetically modified organisms, to clean your water, and then we're going to kill them because they have to die. The concept of kill switch could mean that people are scared. They think, oh no, these microbes must be dangerous. Why do we have to kill them? Is this a negative thing? So at the moment, we're currently looking into implementing kill switches. However, this can be really difficult. Again, for a kill switch that can work based on temperature fluctuation, you can change your temperature in the lab from 37 degrees to 28 and say, hey, there's a temperature kill switch, it worked. But when you've got such a rapid change at 200 litres, how quickly will this kill switch work? You need to think about the environmental impact on this, the temperature controls, is the bioreactor set up for this? And all of the aspects a lot further along the journey than you would normally think when you're just in the lab. There was a really nice paper that looked at changing the light system in a photobioreactor from white light to blue light and acted as a kill switch. But actually, if we were to implement that across loads of different bioreactors, how many have built-in pure blue light, or will it actually kill it? And we also have to think of the costs, the regulations, and hopefully I'm okay for time, but I'll go into a little bit about the regulations now. So a couple of weeks ago, myself and 2 colleagues went to Japan to understand a bit more about synthetic biology regulations, because part of EBIC, we really want this scale up to work. However, we're limited and we can't deploy any of these systems into real drinking or wastewater aspects until we have the UK legislation on our side. These are just a couple of pictures. We went to the UK Embassy, we went to Kyoto, Tokyo University to learn from the Japanese collaborators. And so it looks like there's a lot on this slide, but if I walk you through and just start with the bit on the left about Japan, they have actually got GMOs on the market already. And I think that's pretty fascinating. They aren't readily available in supermarkets. It's actually online, so you can choose to buy. It's not that it's kind of an in your face, here you need to choose to buy these GMOs. It's very much a consumer's choice. And they have some different regulations to what we do. So if there is no foreign DNA, so if you've done knockouts, you can actually treat them separately to GMOs, and they have a different regulation pathway. And then also there's a note that I've put here about labeling requirements. So in Japan, if you have products that have been modified, but it's less than 5% of the total weight of the whole ingredient list, you don't need to label it to say that they are GMO. Whereas in the UK, I believe that's less than 1%. I think it's about 0.9% for each ingredient has to be labeled if 0.9% of the ingredients list is a GMO product. So we need to standardize this somehow and really look into understanding what synthetic biology means, how we can implement it in real world systems and tackle this issue where I think we've been working on synthetic biology for so many years, and it seems like the community are doing such great technology, but there's this real disconnect between the scientists and getting it into market. I think for what we're doing is perhaps slightly harder than the food and agricultural industry, because within drinking and wastewater, it could get into the environment. There could be what we call deliberate release, because we need to make sure that we've got the proper controls in place. Whereas if you're selling a GMO that's plant-based or a food-based, you might have tighter regulations surrounding this. So it's a lot of things that we really need to consider and work on. So take-home messages, don't know why this one's popped up, but just for the SynBio community, I think we really need to have better tools for environmental microbes. I think we've done really well at using model organisms, studying them to the point that we know pretty much everything about them. Whereas when we want to translate this into real world applications, we can't just deploy E. coli into these systems because it will probably die. Even if we make it into the best bug that exists and we've modified it so the strain engineering is great, it can survive in bioreactors. If you have a natural system of wastewater, which has got so many different microbes, prokaryotes, eukaryotes, it's going to be one of those competition aspects where ultimately they could die. I guess we need to do more studies to really understand the difference between environmental strains. But I think that would be a really good way to sort of move forward. And another one is biosafety. This is something within EBIC, the Environmental Biotechnology Innovation Centre, that we're really, really working hard at understanding, especially at scale. This is to kind of raise the awareness of, we need the public on our side. How do we do this? Because they might not like the idea of having GMOs in our water treatment. And then ultimately, if this leads into the ocean, how could this affect the ecology in rivers? How can it affect the ecology? Which are all really good questions for people to have. And we don't have all of the answers yet. So it's just having a community where we can work on all of these to understand and progress the field of SynBio. And I think what we failed to do perhaps as a SynBio community is to think of scale up from the very beginning, because even for myself being a molecular biologist, I never thought of what considerations you needed for a 200-litre bioreactor. But if we want to take the science to that point, we really need to understand how different these GM microbes are going to perform in a flask compared to how they're going to perform when we're scaled up. And even the 300-litre bioreactor at the 200-litre volume is only pilot scale. If we were to move to industrial level, it's like way beyond that. So it becomes a real big challenge for us. But we need to work with the different companies that are out there. And so that's why I've sort of got a little bit more into policy to try and understand more about the regulations, how we can also push these regulations so that we can actually make a bit of a difference. Because at the moment, even if we find out that our engineered microbes work really well at lab scale, we can't always scale up like we want to. And I think the last point is about public trust. When we talk about synthetic biology, a lot of the time it's with our own community, and people within the community of biology are really open. They think it's a really cool technology. And I guess sometimes you have to ask the question, just because we can, doesn't mean that we actually should. And does the science work better than the wild types, or are there alternative methods to cloning, engineering the strains? Could we do adapted evolution? Is that the better solution in order for us to get into this scale up? So I guess there are loads of things that I don't have all the answers, but I'd love to have discussions to hear all of your thoughts. And another thing just to mention is that EBIC have a ECR conference, so Early Career Conference, in September. It's down in Southampton in the UK. I think registration is still open, but I think perhaps the abstract submission is now closed. But it is a really good way to meet other people working in engineering biology, especially for environmental solutions. So if you can, then this is the link. It's on the EBIC Centre website. And this is how you can reach me and find me. But thank you very much for listening.
Thank you so much for the amazing presentation. There's a few questions in the chat. I'm going to ask just one first because it was not towards the end that you're talking about it. When you're talking about the way that we should start talking about synthetic biology with the public, what do you think we're doing wrong? Or how do you think we should approach it? Because you have a lot of experience as a science communicator. So how would you approach talking to someone that might not be very happy with GMOs, etc.?
I think it's, first of all, listening to people. I think the problem can be that we can maybe be pushy in a sense that we know the science, we know exactly what we're modifying. So to us, it's like, well, it clearly isn't going to cause any harm. But I think for a lot of the general public where they haven't got that knowledge, it's not that it's too complicated. I think some people switch off from scientists because they think, oh, you think you're too smart, you think you know everything. But these are real people, family, friends, colleagues, anyone that could just really disagree with it. And I think it's listening to why they don't like it. And if the answer is that they've seen it on the news, then you can open the discussion and really talk to them about it. Because I just don't think people are aware of what GMO means and what synthetic biology is. And when you actually walk them through the process and say, oh, we can target a specific area and only change that, then they might be more like, oh, wow, I thought you were doing something really crazy. Or they always say this playing God aspect. So I think it's first listening and then tackling the points and walking them through things. And I don't believe you should have to dumb down science. I don't really like that term of like dumb it down for the lay audience. It's talk to each person or a group of people in a way that you can relate to them, but still giving them enough of the science. And that's a really hard challenge, to be able to communicate the science in that way.
Yeah, during my IGM project, actually, we had this whole thing that we were just outside and offering cake if people wanted to hear about our projects, but they could only get a piece of cake if they heard what we're doing. And the amount of people that just came just for cookies or whatever, and then in the end they're like, oh, actually that sounds cool. We're trying to make energy out of cyanobacteria, like solar panels. with cyanobacteria. And everyone thought it was a great idea, which was very surprising because I thought people didn't like GMOs. But after they hear that, they're happy about it. So, yeah, I think there needs to be a two-way dialogue.
Yeah. And cake helps, I think. Cake helps with anything. So maybe that's the way forward. Set up stands with loads of cake and tell people to come to a stand. I like that.
Now going to the questions in the chat. We have one that says, how does microbial bioremediation compare per se to other methods, such as via accumulator aquatic plants and algae and subsequent biomass harvesting? Are there benefits to combination of methods supplementing each other for breakdown absorption of compounds?
Sorry, can I read that question or can you repeat it, please?
I can read it. You can also find it in the chat if it's easier to see it written down.
I can't see how to get to the chat. I don't know why.
I can read it again.
Oh, yes, please. Thank you.
How does microbial bioremediation compare to other methods, such as via accumulator aquatic plants and algae and subsequent biomass harvesting? And are there benefits to combination of methods supplementing each other for breakdown absorption of compounds?
Okay, so the question is more about the downstream process, is it? Of once we've bioremediated compared to if you're doing product-based yields of algae?
Yes, that, and if you can mix the two and if there's benefits to that.
So at the moment, we haven't actually scaled up properly to the point of doing that. But what I think can be beneficial is understanding what system you want. So in the introduction that you gave, it was about my work on biofilms. So another aspect which adds layers to this system are that in different treatment processes, you could use membrane bioreactors, and you might want biofilms on your system. You might not want biofilms because it can increase biofouling within your system. We would need to understand what our process is for increasing bioremediation. So, for example, we need our microbes to either secrete enzymes, or if it's an intracellular process, we need to understand what that process is. For the metaldehyde example, we don't actually know if it's an intracellular or an extracellular process. So that's something that we need to look into. But in order to then harvest biomass, again, we're not sure if we need to reseed with microbes or we need to harvest the microbes. But in this case of bioremediation, with the metaldehyde breaks down into a non-toxic compound. So we don't have to harvest those cells and get a yield out of those cells. So the idea would be to have it as a continuous system. Either we can reseed with more microbes. So I don't know how that works in terms of if we need to harvest or a combination of methods, if that answers the question.
Okay. Yeah, I think it does. And the second question is related to whether using a consortia of bacteria of algae would be better or not. So they refer that there's competition in the stream, as you mentioned, and if inoculating a consortia would make it more stable and be beneficial to restrain algal blooms in the phosphate-rich inlets, or if you thought about that.
Yeah, definitely. So it could be that we have to come up with a consortium of microalgae. What some people are doing, and I believe it's as part of EBIC as well, I think it might be at Newcastle University, you really need to understand what each different microbe does within the consortium, because if you have a microbe that breaks down one compound, you need to complement it with one that's using that compound, or so I think that would be quite complex to understand the system, especially if we're engineering one of the strains or multiple of the strains. I think we really need to understand the population dynamic and how they work together. So I think that's slightly out of what we're doing, but it's definitely something that would be really interesting to do and look at these competition assays to see what survives. I guess if we've got a consortia of algae, it could mean that there's slightly even more competition, or it might compete with one other natural microbe, but it doesn't necessarily help our engineered strain. I think there's so many different aspects of it, but it would be really nice to look into communities as well. And was there a second part to that question?
No, that was it.
Yep.
Yeah. There's a question by someone else.
Yeah.
Why did you decide to use a non-model organism?
So the idea for a non-model organism was mainly to see if it would be a better performer in the environment. So if it's a naturally occurring microbe, it could potentially survive better in the natural conditions. But also a little bit of it was to do with the regulations. So I didn't go into it too much, but within the UK, we've got the Precision Breeding Act, and that allows you to use strains that have acquired a mutation that could have been naturally acquired through evolution. So that's mainly for crops. But if you could naturally evolve and get that mutation, it goes down a separate regulatory route. So for this particular environmental isolate, it already has these metaldehyde degrading genes. And all we want to do is add in a constitutive promoter to always express the genes so that we don't get it switched off. And therefore, it might help with the regulation process because we're not adding foreign DNA. It's its own DNA, but we're just overexpressing it. So I don't know if the promoter would be considered as foreign DNA because it's not from a different organism. So it might help with the actual regulation processes to use. But we do think that using a model organism may just be that it doesn't survive in the conditions of wastewater. So we've got different pHs, they can fluctuate. And so I guess it's that balance of understanding, which one we could use. But I think, again, we could try with different model organisms and see if they survive. But I guess it's quite a lot of work to test and engineer loads of different strains to trial. But good question.
I have a question following up that. Do you use directed evolution in your research?
No, we haven't yet.
Oh, okay. Well, it would be quite cool to just mutate the gene you guys are looking at just to make it more efficient as well. I don't know how that looks into the regulatory aspect of it, but yeah.
I don't know either, but I think that would be a really interesting point because if it bypasses some of the regulation of GMO regulations, we could still implement that strategy. Mm-hmm. And so I think it's something we should look into, yeah.
I have one last question, which is a bit related to this regulation bit. Do you, because you're a science communicator as well, do you see researchers worrying about containment enough? I talk a lot with social scientists, and they're always a bit, very interested in those aspects much more than the other bit of it. So, what's your opinion on whether safety and containment is being implemented in current designs? And you have already talked about how you implemented in yours. So,
I think the answer that I've experienced is that we don't, and I'd say that from conversations that I've had going to different conferences and just speaking within the community of synthetic biologists. I think we have containment from perhaps different angles. For our view, it's containment to not enter the system in the environment. But from an IP perspective in industry, people don't want anyone to steal their IP. So they would want to kill the strains before it gets in, so no one can understand what modifications they've done. But I'm a pure biologist through and through. So even before I came into EBIC, I hadn't seen a bioreactor. I'd worked with marine systems. And so it was all very new to me. And things you have to consider aren't just containment at the point of harvesting. And if you want to implement the kill switch before you harvest the cells, it could be, what if there is a leak in your system? What happens if there's a system failure and everything leaks out of your 200-litre liquid bioreactor, all coming out, and where does this discharge to? Does it go to the drains? What happens then? How do you get rid of it? How can you then implement a kill switch when it's all over the floor because it's not in a contained vessel? So things like that, I think perhaps haven't been considered. How do you change the temperature in the room if your kill switch is a temperature controlled one? It can be difficult. Or if you need auxotrophy, if you need to add in a supplement of something for the strains to grow on or kill them, if there's a leak, what do you do in that case? Do you just add things randomly? So again, I didn't consider these things. I just thought, if your system's always working, you can just add in what you need to add or change the physical parameters, and then we can discharge. But I think what I'm understanding is that people aren't satisfied with a one-level containment strategy. So you need physical layers, you need you also need maybe the chemical layers or biological layers. And so where does the line get drawn? I think as a community of synthetic biologists, perhaps we don't consider that as much. And so I don't know if we need to put in more research within the biocontainment strategy, because most of the technology that we've got is great, but it can't be implemented without a really great system in place. I don't know what your experience or other people attending have experience with this. It'd be really great to know, especially if you speak to social scientists. We have a few social scientists on EBIC, so we're trying to have sort of several different aspects of the project to make sure that we're engaging with the public, with policy, to really make sure that it doesn't just stay in the lab and we can implement this in the future.
That's the great thing about hubs in the UK, right? They give a lot of money and you can hire a lot of different people doing different things, but always related to the same topic.
Yeah.
I agree completely with you. And even when I interact with policy and social scientists, it just feels to me that the wet lab scientists are doing their research sometimes in a bit of a vacuum, like they don't know what's going on outside. But at the same time, being a researcher is already so tiring. Should we know everything, Maybe we should just have more interdisciplinary teams, and maybe it'll take a bit of a longer run to get anything done, because we'll disagree with each other. But maybe at the end, the product will be much more useful for humanity.
Yeah.
But yeah, these things are they're very interdisciplinary, and it just gets very difficult to be an expert in absolutely everything. And trying to get everything done. So,
and I think funding, there's within the mission hub, like you said, so EBIC's part of a mission hub in the UK. I think we've got 4 different mission hubs that allow people to research different areas. And this one's all about engineering biology for the environment. And we are able to hire social scientists to work on these issues. But I think a lot of other funding calls don't necessarily include social scientists. So maybe it's finding the funding avenues where we can have that, or making sure that when we have meetings like this, that we're engaging with the social scientists and making it an open space for them to attend as well. But I think, a lot of it's down to funding and understanding what we can do.
And I think one of the main complaints also here is how the social scientists, they get involved a bit too late in the project. Like in the hub, in hubs is different, but in other funding calls, like you said, right? You interact with the social scientists after you have written in the grants, like, I'll do, I'll ask people, the public, about what they think, and then they contact the social scientists instead of contacting the social scientists when they're starting thinking about the projects. Yeah. And then projects just die off because there's just they can be commercialized, and now the government loves that. Exactly. That you can make money out of your research. So things get a bit lost in the vacuum as well.
It's true. So the recent trip to Japan was through Cranfield University and engaging with policy. So we met with some social scientists to talk about the Japanese public's view and perception on GMOs. And what's nice is they have some really nice case studies or surveys. There was one particular one on cultured meat. So it was looking at, what are your views on cultured meat? Would you try it? And the disconnect between people that are like, I might try it, to people that actually would go out just because you could try it. Do you actually want to try it? And then understanding, what does that mean if it's cultured meat? vegetarians have it? Because is it still classified as meat? And so there were different public perceptions on that. But another thing that we need to probably get better at is having more conversations within policy, but with different countries. If we make a technology here and it's not standardized, like in Japan, completely different labeling system. If we made a product here, everything's different, the different regulations for when you're importing and exporting, what are your processes. I think one thing that we did find quite useful is one of the companies does like a business to business model. That's where they can produce the engineered product that they are and give it to another much larger company, and then they can go through the regulation process to get it on the market. Whereas I think a lot of startups, in particular ones that I know in the UK, might be trying to do everything. They might try and make a new product and then get it to market without having non-GMO products on the market. And then if that fails, then the company is ultimately going to fail because they haven't got a backup. So I think it also depends on what our end goal is, who our consumer is. Are we making products, or are we, I guess what we're doing with Anibic is kind of producing a service. Who are our customers? How are we going to make it financially viable? And so again, coming to the point of, do synthetic biologists think about biosafety enough at the start? I think you also need to think about what is the purpose and who is your customer.
Yeah. Unfortunately, I have to cut off here because we have a panel in 7 minutes. We'll be a bit late already for that. But I wish we could continue. Thank you so much for coming, and thank everyone else for coming as well. Follow Yasmin on socials if you want to hear what she's up to. And yeah, go to our panel in, let's say, 10 minutes because we'll be a bit late.
Perfect. Thank you so much, guys, and thank you for having me.
Bye.
Bye.